So we were talking about fluid mosaic model, and you remember that from last year? Why is it fluid? What makes it fluid? It's wherever we left off yesterday. Okay, so what makes it fluid? Cholesterol was one. What was the other few things? The unsaturated is the one with the K. So unsaturated fatty acids, cholesterol. What's the third thing? Movement of the phospholipids. What kind of movement do phospholipids do? Right, so lateral is a lot more common. Flip-flop, a lot more rare, but it still happens. Okay, here we have this question. Let's see what it says. It says a student proposes 6 models of a hypothetical plasma membrane. Describe the model that best represents the structure of a plasma membrane in a non-aqueous environment with a cytosol environment similar to living cells. So the cytosol is similar to what we have, but the environment, what's surrounding the cell, is going to be non-aqueous. So which one of those models would make sense and why? Right. Non-aqueous, it's not, there's no water there. No water. Non-aqueous is no water. Well, you tell me, if it's non-aqueous, there's no water. If there's no water, which side of the phospholipid is going to? Both sides. It says the cytosol environment is similar to living cells. So, well, A is what we have, but that's not non-aqueous, because what do we know about the heads? It wants to be near water, right? Well, B is only one-sided. The head still doesn't want to be in non-aqueous, and it says the cytosol is similar to what we have. That's not non-aqueous. C is only facing the hydrophobic to the cytosol, but what do we know about the cytosol? It's probably aqueous, right? Is it your cell aqueous? Yeah. So, how about D? What do you know? What do you see with D? Right. And then we see E. Now, why not D and why? No, stop. Okay, so let's think about this for a second. D, the heads go towards the but you still have heads facing the phospho. So between D and E, but by the way, D and E are the only ones that make the most sense, right? So between D and E, why is it E and not D? Because all your questions, I don't know if you kind of paid attention to it on your quiz, but a lot of the questions are going to give you 2 choices that really are incorrect, and then 2 choices that make sense, right? In this case, both D and E make sense. But there's something a little off with D that doesn't work very well compared to E. What do you see there? Okay, so the heads are interacting with the tail, shouldn't be doing that. Okay, so if that's the case, then does That's not going to work, is it? Because if it's aqueous, the cytosol is aqueous, it does not want the tail, it wants the head. And so D doesn't work because the heads and the tails are interacting with each other. And F doesn't work. So you're left with, it must not be a bilayer, it must be a single layer. Okay? So that's why it's E. All right. So phospholipids spontaneously form a bilayer. What does it mean for it to form a spontaneous bilayer? The minute it comes together, the tails face each other and then it forms a shape where the tails are all together and the heads are facing the aqueous environment. There are proteins in there that are embedded, right? The integral proteins that are used for transport and many other things we're going to see. Then you have peripheral proteins that are loosely attached that are also going to have different functions. Yes. No, the bilayer, the minute you dump phospholipids into an aqueous environment, it makes a bilayer. It tries to, yeah, so what it does is the minute you put it into an aqueous environment, the tails all come together and try to stay away from the aqueous environment. What would happen if you threw the phospholipids into a non-aqueous environment? How would they orient themselves? Tails out, all the heads would come together into one area, trying to stay away from the non-aqueous environment. Okay? All right. On that note, now we're going to talk about permeability. All right. So the cell membrane is selectively permeable. We've said this before. What does that mean? It only allows certain things through. Why does it do that? Right? You want a certain number, you want a specific concentration, and you don't want it freely to take in everything. Right? You don't allow everyone into your house. Right? That just because somebody knocked on the door doesn't mean you allow them into the house, right? It's if you're selecting the individual that comes into your house, the cell selects the molecules that come in and out. So selectively permeable, small non-polar molecules pass freely. What are small non-polar molecules? What do you know that is a small and a non-polar molecule that the cell would probably need, and it would go in and out really, really easily? Oxygen, carbon dioxide, small, and they're non-polar, right? What about water? Oh, but don't we need water to go in and out as it pleases? Is that an osmosis? But then what happens? There must be something that membrane has, right? It's called aquaporins, which we'll talk about. Okay, so nitrogen, oxygen, small, they're the tiniest ones that are just going to freely go through. Carbon dioxide is a little larger, so there's some polarity with it, but it's still considered overall a non-polar molecule. What if we want something to go through, but it's hydrophilic or it's too large? So this is where you need facilitated diffusion, right? Hydrophilic substances such as large polar molecules and ions cannot easily go through. Hydrophilic substances have to move through a channel protein, and the channel protein has to have a hydrophilic substance. Without that, it's not going to be able to go through. That's called facilitated diffusion. First of all, let's define diffusion. What's the definition of diffusion? What is the direction of molecules? Which way do they go? High to low concentration. So where you have a lot of molecules, so where you don't have a lot, it goes from high to low. Okay, great. And then? It's still high to low. What does it mean permeable for it to go through. So for that reason, it has to go through a channel. Okay? All right. You have carrier proteins. What's the difference between a carrier protein and a channel protein? They're both integral proteins, but there is a difference, and you can kind of see that difference on this picture. What is that difference? Right. Channel protein is literally just a channel. It's a hole in the protein and things are flowing through it. The carrier protein grabs it from one side and dumps it to the other. So it grabs it, let's say, from the outside environment and then throws it into the inside environment or vice versa. Okay? Not necessarily. If it's down its concentration gradient, not necessarily. All right. If it's a plant cell and some bacteria, along with the plasma membrane, it has another structural boundary, right? Which is the cell wall. And the cell wall protects and maintains the shape. It prevents against high pressure, and it helps plants stand up against the force of gravity. So it gives plants a lot of structure, and they are still permeable. So they have pores where water can easily go through. They're actually connected with something called plasmodesmata. And that plasmodesmata is important because it allows for flow from one cell to the other. Since remember the cell wall is a rigid structure made up of cellulose, right? And it needs to allow water, nutrients, waste easily to pass through, and it can't do that unless it has some kind of opening. Okay? The cell wall is just an extra barrier that surrounds the membrane. Okay? So plasmodesmata is found in front cell walls. It's an opening, and it connects one cell wall to the other, allowing for things to easily flow. Okay? You with me so far? Yes. Yeah, great. Yes, we can. And we know already that the animal cells don't have cell walls. They have to have energy from low to high, and in some cases, under certain reactions, they might need energy in order to flow through its body. But for the most part, high to low is a downhill process, right? So it does not require ATP. Unless there's an enzymatic activity. Okay? Questions on that so far? Alright, so plant cell walls we already know are cellulose, but there are different kinds of cell walls that exist. Fungi have chitin, it still gives it structure, it's still a polysaccharide, and then prokaryotes have something called a peptidoglycan, which is still a polysaccharide with amino acids. So all of those are cell walls, and they're all complex carbohydrates, and it's important because it gives it